Hi my dear students, today we will follow and finalize section 2 chapter 23. In this video we will explain enzymes, their functions and denaturation of enzymes. But before we start, we have to highlight on the previous knowledge. Number 1, we are studying biological compounds which are carbohydrates, lipids, proteins and nucleic acids. Number 2, we finished carbohydrates, lipids, and today we will finish proteins. Before we will start enzymes, we have to understand that nitrogen accounts for about 15% of the mass of the protein molecule. Most proteins also contain sulfur, some contain phosphorus or other elements such as iron, zinc and copper. And as we discussed before, proteins have many important catalytic, structural, regulatory and antibody defense functions. Like keratin is the main component of hair and fingernail. Number 2, enzymes catalyze biological reactions. Number 3, hemoglobin carries oxygen in the blood. Number 4, insulin regulates glucose levels in our body. Number 5, antibodies protect the body from foreign substance. So now we have to understand that enzymes are part or form of proteins. We have to differentiate between catalysts and biological catalysts. Catalysts are chemical compounds that accelerate the chemical reactions and decrease the activation energy and are not changed during the reaction. But biological catalysts are enzymes, which are protein in nature and active inside living cells to enhance the biological reactions and decrease the activation energy too. So, proteins are as enzymes, and enzymes is a protein that catalyzes biological reactions. Enzymes make up the largest and the most highly specialized class of proteins. Most enzymes are water-soluble globular proteins. The amino acid side chains and the three-dimensional shape of enzymes play a very important role in the enzymatic activity. So return please to the last video yesterday. So an enzyme also does not change the amount of the product that is formed in the reaction. It only decreases the time it takes to form the product and decrease the energy required. Then, enzyme catalyze both decomposition and synthesis reaction, so it will act on both sides, the forward reaction and the backward reactions. Enzymes are very efficient, so very small amount of the enzymes can enhance a huge amount of the reactants. So a single molecule of carbonic anhydrase enzyme can break down 36 million carbonic acid molecules in 1 minute. Enzymes are characterized by a specific character, which is a specificity. Enzymes are very specific and often catalyze just a single reaction. So for each reaction we have only one enzyme for this reaction. So enzymes act by binding to a specific substrate molecules. Also, the shape of the enzyme is such that the substrate can fit into the enzyme at a specific part of the enzyme molecule called the active site. So the substrate can only connect with these active sites. Combination between the substrate and enzyme will produce enzyme-substrate complex. This model of enzyme action is called the lock and key model. You can see this slide. Now you have the substrate and also you have the enzyme, and you can see the active site and the surface of the enzyme. The substrate will connect with these active sites then to form the enzyme substrate complex. Then this enzyme substrate complex will hydrolyze to the product, and the enzyme again return to start another reaction. And as we mentioned before, enzyme is a type of a catalyst, and it is a biological catalyst, so it increases the rate of the reaction or accelerates the reaction and decrease the activation energy or decrease the energy required to start the reaction. Let us see these curves. This figure explains the reaction between reactants without the enzymes and the same reaction with the enzyme. See here, the amount of energy required decreased, so the activation energy with the enzyme decreased. So you can see this area with using of enzyme and this area without using of the enzymes. Now we have to explain factors that affecting the enzyme activity. We have two main factors affecting the activity of enzymes inside our body. The first one is the temperature, the second one is pH of the media. And you have to understand that all our biological catalysts inside our body will be activated, or the maximum activation of these enzymes is at 37 Celsius and the neutral pH of the blood. If we have any change in the body temperature or the blood pH, that means denaturation of the enzymes will happen. So we have a new expression which is denaturation. Denaturation is a change in the protein's characteristic three-dimensional shape due to change of its secondary, tertiary or quaternary structure. Why and how? Why? Because changing of the temperature and pH or one of them will make denaturation. How means the three-dimensional structure will change, so the site of activation or activation site that the substrate will connect with the enzymes will be denatured or changed, so the enzyme now is inactive. And we have example like cooking of eggs. As we know, the pH of our blood is ranged between 7.3 and 7.4. If the pH changed, decreased or increased over or below this range, the enzymes will be deactivated. The last part of this section is pH and enzyme activity. We have to understand what's the meaning of pre-enzyme. Pre-enzyme is the inactive form of the enzyme. Like in our stomach, we have a cell wall of the stomach, and this cell wall is responsible for production of some enzymes as a pre-enzymes or inactive form of the enzymes like pepsin and trypsin. Pepsin will be active directly when it will be contact with the acid inside the stomach. So it will change from the pre-enzyme to the active form pepsin, which is responsible for digestion of protein and change it to polypeptides. The second enzyme which is released from the wall of the stomach is trypsin, but trypsin has specific media. It will be changed from the enzyme to the active enzyme in the alkaline media, so it will be active only in the small intestine to change polypeptides to amino acids, which are the smallest units will be absorbed to our blood. Now we finished our section in chapter 23. Thank you. See you later.